In this video, we'll focus our attention on deoxyribonucleic acid or DNA. We already know that the primary structure of DNA consists of a sequence of nucleotides linked together via phosphodiester bonds. So what does the secondary structure of DNA look like? By the time scientists started looking at the structure of DNA, they had already worked out the structure of nucleotides. They knew that DNA polymerized through the formation of phosphodiester linkages, and they also knew that the molecule had a sugar phosphate backbone. With this information, they now wanted to find out how the sugar phosphate backbone is arranged in three-dimensional space. It is from the work of many scientists that we know what we know about DNA today. One of those was Erwin Chargaff, who established two empirical rules for DNA. One was that the total number of purines is equal to the total number of pyrimidines. And second, the numbers of A's and T's are equal, as well as the number of C's and G's. Another pair of scientists, Rosalind Franklin and Maurice Wilkins, calculated the distances between groups of atoms in the DNA molecule. This established the width of the DNA, which was 2 nanometers, and the distance between two adjacent nitrogenous bases stacked one on top of the other, which was found to be 0.34 nanometers. They obtained this information by bombarding DNA with X-rays and analyzing how it scattered the radiation. This technique is called X-ray crystallography. The data for the scattering patterns showed that 3 distances were repeated many times. This led them to conclude that DNA molecules had a regular repeating structure. The pattern of the X-ray scattering also suggested that the molecule was helical or spiral in nature. Using this information, James Watson and Francis Crick started building physical models of DNA. They tried adding a purine-purine base pair and found that there wasn't enough space given the width of DNA. They tried a pyrimidine-pyrimidine pair, which turned out to result in too much space between the nitrogenous bases on opposite strands. When they tried a purine-pyrimidine pair, the distance was just right. This led them to conclude that purines must base pair with pyrimidines on opposite strands of the double helix. We know now that this interaction occurs via hydrogen bonding between the nitrogenous bases on opposite strands of the DNA. So we now know that DNA is a double helix composed of 2 strands. You can see the sugar phosphate backbone of the 2 strands. So there's one strand, and then the other strand runs in an anti-parallel direction. The strands are composed of individual units called nucleotides, and these nucleotides in turn are composed of a nitrogenous base, a deoxyribose sugar in the case of DNA, and a phosphate group. Let's look at one purine and one pyrimidine. Let's start with a purine. This nucleotide is composed of a nitrogenous base called adenine, a deoxyribose sugar that is attached to the nitrogenous base as well as a phosphate group. Let's look at T here. This is the base thymine, which is attached to deoxyribose, which is then linked to a phosphate group. Now these nucleotides come together to form phosphodiester linkages, which are bonds that form between the phosphate group of one nucleotide and the deoxyribose sugar of the next. So here's the nitrogenous bases A, C, G, and T, and the sugar phosphate backbone, which consists of alternating phosphate groups and deoxyribose sugar. The opposite strand also has the same structure and is anti-parallel to the first strand. The nitrogenous bases on the two strands interact with each other via hydrogen bonds. These hydrogen bonds stabilize the double helical structure of DNA. So to summarize the secondary structure of DNA, it consists of two anti-parallel strands that wind and twist around each other to form the double helix. The sugar phosphate backbone is exposed to the outside and the nitrogenous bases face the interior of this double helix. Purines always base pair with pyrimidines. A always base pairs with T, and G always base pairs with C. A and T interact via two hydrogen bonds, whereas G and C interact via three hydrogen bonds. These bonds help keep the double helix stable. Another thing to note is that the double helix also has two unequally sized grooves called the major groove and the minor groove. Now that we've looked at the structure of DNA, let's look at its functions. DNA contains biological information and can store and transmit this information from generation to generation. It carries information required for the organism's growth and reproduction. The language of nucleic acids is contained in the sequence of the bases. DNA also has the ability to replicate. The primary structure serves as a template for the synthesis of a complementary strand. The sequence of bases provide the information required for a copy. The complementary base pairing provides a simple mechanism for DNA replication. Each strand can serve as a template for the formation of a new complementary strand. So how does this process work? First, the two strands in the double helix separate. Each of the two strands then serve as templates for building new complementary strands. As new nucleotides are added to the growing new strand, polymerization helps form phosphodiester linkages between the nucleotides. DNA is a very important molecule within biological systems because its stability makes it a reliable store for genetic information. Since it is stable, it is less reactive than ribonucleic acid. It is more resistant to chemical degradation. Because DNA is such a stable molecule, it makes a poor catalyst, so it does not play a role in increasing the rate of chemical reactions. Since it does not have catalytic properties, biologists think that the first life form made was RNA and not DNA. This concludes our video on DNA. At the end of this section, I would like you to be able to answer the 3 questions listed on this slide. Thank you.